cleft right there. This whole area would be a neuromuscular junction or just an example of a synapse. But there is no direct connection. First clicker question of the day. We got an A and a B choice up here. Maybe you're wondering what that's about. And just to make sure you're following along so far, and I think most people did catch this, which direction is that signal coming along the axon, or how's it going along the axon? If you think it's going from here, the axon's the yellow, and if you think it's going from here to here, you would pick choice A. And if you think it's going from top to bottom, you'd pick choice B. So you think the arrow should be pointing down. If you think choice B is correct, you think choice A is correct, you would pick the one where the arrow is pointing up. Okay, let's see, just to make sure you got this information. Okay, you guys picked nearly all B, that is correct. B is the correct one, so it's going down the length of that axon. So it's going from axon to to muscle cell, muscle fiber. Rihanna?
Okay, I know we don't need to know this yet, but is the synaptic inbulb part of the terminal button?
Yes.
Okay.
Yes. In fact, that might be just another equivalent name for terminal button. Yeah. Let's take a look at the next picture. Here's maybe a closer look at this. So now we're looking, this is the gap between our between our neuron here and our muscle cell here. So this gap is occurring. Well, how do we get this information then from one cell to the other, because they're not directly connected? And the way it moves down that axon is mostly electrical. But you know we have to have connection between things for electrical systems to work. We've got something plugged in here to an outlet. It actually has to be in contact with the metal. Various metal. The plug has to be in contact with the metal of the outlet. If you hold it out away from the plug, it doesn't work. You can't get a transmission. And these are not actually touching. So how do we get this information from the neuron, the nerve cell, over to the muscle cell? And we do that by certain chemicals called neurotransmitters. So what's going to happen is we have these neurotransmitters. And on this image, these little red symbols here indicate neurotransmitters. These are chemicals. They are being released from the synaptic inbulb. They're traveling across this synaptic cleft, and they're interacting with the muscle cell. They're going to trigger something to happen on the muscle cell. They're a communication device. How do you think the word acetylcholine relates to neurotransmitter? Any idea what that is? Acetylcholine.
I'm guessing the first part means acidic.
Okay, that's part of it.
Does the first part of the word mean acid?
The name of the neurotransmitter involved here is acetylcholine. Acetylcholine is a kind of neurotransmitter. So there's different chemicals that could be neurotransmitters. Might be in different parts of the body depending on different neurons. The one that's involved at neuromuscular junctions is acetylcholine. This is the one that's actually going to trigger a muscle cell to contract. Can you guys think of any other neurotransmitters? That's something critical you have to know right now. What was that? Dopamine, yeah, in the brain, for example.
Serotonin?
Serotonin in the brain, yes. Epinephrine, norepinephrine. Melatonin? It might be able to act as a neurotransmitter too, I don't know.
Doesn't lactic acid play a part?
Melatonin works as a hormone for sure. So it's a hormone, a blood-transported chemical. It might work as a neurotransmitter. That may be beyond what I know. Josh?
Isn't lactic acid part of the muscle contraction?
Lactic acid you're thinking of?
That's what
It is a end product of certain kinds of muscle contraction, but it's not a neurotransmitter.
Okay.
And it is on a PowerPoint we're coming up ahead, so we will get to lactic acid.
I want to say one thing, but I'm thinking that actually is a drug that makes something else.
What were you thinking?
Like morphine or something
Morphine is going to mimic something else, yes. Yeah. Yeah, it's not something that we would produce directly in our own bodies, though, as a neurotransmitter. Okay, anyway, you don't have to write all those transmitters down. The only thing I want you to know for now is the chemical that moves across the synaptic cleft is a neurotransmitter, and the kind of neurotransmitter used at this junction is acetylcholine. So that's how those words are related. It is going to interact with receptors which are on the muscle cell, the muscle fiber. The green blobs down here represent receptors. That is, they are particularly sensitive to acetylcholine. When they get bound with acetylcholine, or when acetylcholine binds to them, it's going to trigger something. Okay, let's take a closer look at this picture here now on the bottom right just for a minute. I think we've got all the same things outlined. And again, you see the close-up view, so the same terms here in bold and green. And it looks like I've got an A and a B again. I don't even quite see the things, but I am asking what direction the neurotransmitter moves. AKA, what direction does the acetylcholine move? And again, your choice is, if you think A, it is going to be moving upwards, the arrow should be pointing upwards. If you think choice B is correct, you are basically saying that the acetylcholine neurotransmitter is moving downwards. Just make sure you caught what I said earlier. Make sure we're all caught up here on this before we move on to the next thing. Which way is acetylcholine moving across that synaptic cleft? Here, this space in here is the synaptic cleft. And you guys chose on this. Let's see, mostly B. Okay, very good. Everyone's following along.
B.
Yes, so acetylcholine is moving from the neuron, the synaptic involved with the neuron, down to the muscle fiber, down the muscle cell, where it interacts with the receptors. Once it interacts with the receptors, that sets up something called a muscle action potential, which is our next slide here. Orientation about what we're looking at. Yellow here is our axon and our synaptic inbulb. There's the cleft right there, the synaptic cleft. You see it better as a gap. We've got our little red dots here that are neurotransmitters. Our receptors are down here. We can see this ACH receptor down here. And now we are going to generate this thing called a muscle action potential. These red arrows indicate a muscle action potential. What it is, it's an electrical wave moving across the membrane, the sarcolemma, the muscle cell's membrane. So it's an electrical wave moving across the sarcolemma. And more or less, it initiates at a point and then spreads outward. So like you throwing a rock in a pond, you throw a rock in a pond and you get these waves that spread outward from that. The same thing happens for this muscle action potential. And it will automatically propagate from there. It requires no additional energy. It's sort of triggered, it opened up, and it just sort of self-propagates over the whole surface. It propagates over the whole surface and down deep in the T-tubules, so that events that occur at the surface get down deep as well. This is going to trigger these calcium release channels to open. So remember, I don't remember what color I had. I think I had those, the blue ones on the artwork that I drew earlier that opened up and allowed the calcium to rush out. That electrical wave is what's going to trigger these to open. Calcium comes flooding out, and that triggers muscle contraction. Questions? Kind of some complex topics. You may have to think about that. Read some more information in your textbook, or perhaps look in your look at some of the Wiley Plus animations as well. Okay, moving on from that one now.
I have a question.
Yes.
So do the receptors, do they trigger the muscle action potential?
Yes. When acetylcholine binds to them, that causes a muscle action potential to be triggered.
Okay.
As long as acetylcholine is bound to them, you get continual muscle contraction, or at least, sorry, that muscle action potentials keep getting generated. Okay, just make sure again that you are paying attention. One more clicker question, just to make sure everybody's following along so far, because you kind of have to know the first steps to get the next steps. I just want to make sure everybody's got this. Where is the calcium release channel on the diagram above? So that last item on the list says calcium release channel. Did you guys make sure that you catch where that calcium release channel is at? Just a few seconds on this one, you should be able to catch this one pretty quick. Okay, you guys chose D. D is the correct place. This is the calcium release channel. Now we're linked back up to what we talked about before. So in our big picture idea now, we can talk about how muscle action, another animation I try to make, try to explain what's going on. Essentially this is an electrical wave, and think of electrical wave as having positive and negative charges associated with it. So the way our membrane is originally set up, the green is a cell membrane, in this case it's the sarcolemma, the blue are proteins along this, and the way this is normally set up is there's more sodium outside than there is inside. And what we're going to do is this electrical wave is going to trigger one of these proteins to open, and that's going to allow sodium to rush in. The next one is triggered by electricity, and as the voltage changes, the next one opens up automatically. The next one opens up automatically. The next one opens up automatically. And thus it self-propagates down the whole length of the membrane. So if we put this into animation mode here, we're going to activate it and see what happens. Once the first one starts, the whole thing self-propagates. So as we start out, they just simply come rushing in, and it propagates down the whole length of the axon. And that's kind of what we have, this idea of this electrical wave that moves across the membrane. And it's changing voltage, because what we had before was primarily positive charges on the outside. Now most of the positive charges on the inside. We'll explain a bit more in detail about how membrane functions work, maybe with the neuron system. But this is enough for now for the muscular system. Now our quick overview. We now have this idea that impulses come down the axon, triggers neurotransmitters to go across the synaptic cleft. They bind to the receptors. That triggers the action potential. Calcium gates are opened, and now linking back what we did before, calcium goes down to our thin and thick filament area, our myofibrils. They bind to that troponin tropomyosin complex. Active sites are exposed on actin. Now myosin can bind to actin because ATP is always present. Now we get muscle contraction, and we get the filaments slide past one another. Josh?
Might be skipping ahead, but aren't the neurotransmitters recollected by the synapse?
Yes, that is coming up on just a couple slides.
Okay, cool.
In fact, that may be partly of the next slide. We're getting ready to think about how is this cycle turned off. And key thing on that is what I've got outlined in this black box, this kind of statement that says acetylcholinesterase in synaptic cleft destroys acetylcholine. ACH is the abbreviation for acetylcholine. So another muscle action potential does not arise. And we are going to divert now back to Wiley+, and it's going to tell us a little bit about acetylcholinesterase, and we'll see if you guys figure that out from Wiley+ here now. So let me catch on to where we were at here. Okay, we don't want that one, we want to go to here. And hang on just a minute, let me find my correct one. There's just two of these that I want to show you. Neuromuscular junction. We'll do the first one as kind of an intro here.
A nervous impulse, also called an action potential, starts from the brain or spinal cord to signal skeletal muscle cell contraction. The action potential moves along the membrane of a motor neuron toward the skeletal muscle cell. The junction of these two units, the somatic motor neuron and muscle cell, is called the neuromuscular junction.
Okay, just a quick overview there, and let's go to transmission. Let's go to this one right here. Like this one.
As the action potential arrives at the neuron's synaptic end bulb, it causes a depolarization of the motor end plate. This change in voltage opens calcium channels and allows calcium
This is a different calcium channel, and I didn't cover this, trying not to overburden you with too much information at once. It's another place where calcium is involved, but we didn't talk about this. This is not the same as the calcium in the sarcoplasmic reticulum. It's another place where calcium is involved. You're getting the idea about why blood calcium homeostasis is so important. Calcium is a very important ion.
To rush into the neuron. This influx of calcium ions causes the synaptic vesicles in the neuron to fuse with the membrane.
So you see them going across the synaptic cleft. They're going to fuse up with this green thing here. There's our receptor. The green thing is our receptor.
Thereby releasing the neurotransmitter acetylcholine into the synaptic cleft. Acetylcholine triggers the opening of sodium-gated channels, flooding the interior of the cell with sodium ions. Increased sodium ion levels generate a muscle action potential that travels along the sarcolemma.
Okay, so that's a little bit of review of how that works. We saw the neurotransmitters moving across the membrane. I thought this had something about acetylcholinesterase. It didn't, so we'll just go on the next slide when I talk about acetylcholinesterase. So, oops, wrong one. Clicked on the wrong thing here. Hang on just a minute. Back to where we were at. Okay, and we'll go from there. So let's talk about acetylcholinesterase. There is a chemical, and it's an enzyme. It has ASE.
Just real quick to make sure.
Sure.
So the calcium release channels, the calcium is flowing out and the sodium flowing in?
Yes, but not at the same place.
Okay.
The sodium is moving in and out right here at the sarcolemma, this outer blue line. That is basically where this membrane potential wave is moving. But what's going to happen is, as it moves, it's going to cause an electrical change inside to outside over the whole way. You're going to cause a change inside the cell here of ions. And those ions, that change in electricity, more or less, voltage, is going to trigger these to open.
Okay. And that's when the calcium goes
That's
out.
when the calcium floods out, right? Acetylcholinesterase. It ends with an ASE, which means it's what kind of a molecule? It's an enzyme. So this is an enzyme. So acetylcholinesterase is an enzyme, and it's an enzyme that breaks down acetylcholine. In fact, it's always present in that synaptic cleft to a certain degree. It's in the synaptic cleft. And it fairly rapidly breaks down any acetylcholine that's present. I think Josh mentioned about reuptake. There are some neurotransmitters that actually can get reuptaken, I'm not sure of the phrase there, back into the synaptic involve, but not this one. This one actually just gets destroyed by something called acetylcholinesterase, constantly being destroyed. So if you want a continual muscle contraction, you have to keep pumping acetylcholine out into the cleft. Because once you stop pumping acetylcholine out in the cleft, acetylcholinesterase immediately destroys any that's left in there, and the contraction stops. You have no more muscle membrane wave that occurs across, no calcium release channels open, and you stop muscle contraction. So acetylcholinesterase, again, is an enzyme that breaks down acetylcholine in that cleft. And we need that. I mean, we need that to make sure that muscles, cells don't continually contract, don't over-contract.
So if you've got a muscle cell that's twitching, is that kind of what's going on?
Possibly, you're getting muscle contraction that's occurring there, whether that's caused by an inappropriate signal from the nervous system or exactly what's going on. It may not be a problem with acetylcholinesterase. It's probably a problem with the nervous system. Actually keep telling it to contract for one reason or the other. That's a good question. Maybe I'll have to look up what's the actual chemical level of getting like a muscle twitching, continually twitching. Oh, okay, now we're getting applications to kind of scary things here as we move on from this. There's a number of chemicals that are used to kill things. Parathion, malathion, some other ones. These are insecticides. These are neurotoxins, basically what they are. The same thing also for most nerve gases. Nerve gases, it doesn't take very much of these to be lethal at all. And one of the characteristics of action for both of these Now, by the way, parathion is toxic, a little more toxic to insects than humans, but it's quite toxic to humans as well. That's why in a lot of cases, some of these have been banned, these insecticides, these neurotoxins. Some of them you have to have a certain license to use, or they have very much restrictions on sale and use and things like that. One of the characteristics of people or individuals who have been poisoned by either these insecticides or by nerve gas is they have these spasmodic continued contractions. Their muscles cannot relax. And the reason for that is these particular toxins or insecticides are acetylcholinesterase inhibitors. Does everybody kind of get what that implies? You do not produce acetylcholinesterase, or there's not enough of it there. So there's no acetylcholinesterase, and that means muscles are in continual contraction. Why would that be a problem if you're a person?
They would break down over time, wouldn't they?
Well, it wouldn't. Something else would happen pretty quick before
your heart.
you might have that. I mean, you might have painful contractions,
When your
but
heart give out?
well, that could be to a certain
Do
degree.
you run out of calcium?
Something to do with your diaphragm and breathing muscles.
Oh,
Now remember, breathing, you have to contract and relax, right? You have to contract, then relax. Contract, relax.
can't
If you
relax.
continually contract, you're not breathing. You have to have contraction and relaxation. So one way that you could die by having a problem at the neuromuscular junction is there is no acetylcholinesterase. Your muscles are continually triggered to contract. You can't breathe because they're in continual contraction. It's called spastic paralysis, for example.
so is that why when you spray an insect, their legs just kind of go crazy?
Probably, yeah, probably.
Cool.
On the other hand, there's another way that things can be killed or injured by problems at the neuromuscular junction. Other poisons work a different way. These particular poisons create what's called flaccid paralysis, where the muscles don't contract at all. They become very limp. In this case, we've got botulism toxin. You've heard of the word, or heard of the disease botulism before. It's actually a toxin secreted by a certain kind of bacteria. And we got the bacteria name up there. You don't have to know the bacteria name. But it secretes a certain kind of botulin toxic, and it prevents release of acetylcholine. So it actually is produced here in the synaptic end bulb, but never transports across the synaptic cleft. So you don't get any signal to contract. The muscles stay limp all the time. And again, you don't breathe and you die, this time from relaxed paralysis, from flaccid paralysis. And that acts right at this level. Curare. This is a poison that South American Indians have used for hunting various kinds of monkeys or other animals. They use a couple of different things to actually tip darts with. One of the things they use is curare, and it's a South American plant. They also use poison frogs as well, but this is something different. Curare, anyway, it blocks ACH receptors. So what you have is ACH being produced. It moves across the synaptic cleft, but there's something in the way. They can't get to the receptors because they're blocked up from curare compounds. And again, you do not get an action potential across the muscle membrane, across the sarcolemma. You get no muscle contraction. You get flaccid paralysis, and the animal dies. In this case, if they're hunting monkeys, the animal dies because the muscles are limp and relaxed. And it works pretty good if it's a monkey too, because you don't want the monkey to spasmodically grip the limb, up 100 foot up in the tree. You want it to release its grip and fall down where you can get to it. You don't have to climb a 100-foot tree to get the monkey there.
Poor monkey.
Isn't that like Botox?
Yes. That's exactly what happens there. Yeah.
How does that prevent wrinkles?
It actually relaxes muscles. It's in lower concentration, but actually relaxes muscles in the face.
Is that why when someone's had an excessive amount of Botox, they can't move their face? It's just kind of stuck like this?
Maybe. Don't have a lot of familiarity with that myself, so that may be true. I don't know anybody who's had that treatment, so I haven't really noticed that one way or
Sister
the
had it
other.
one time.
Yeah, but it actually, the wrinkles in part are caused by overcontraction, maybe not overcontraction, but you can sometimes reduce the wrinkles a little bit by relaxing those muscle contractions. I think we'll talk about, I think I've actually got some slides on Botox a little bit later with the nervous system as well. All right, might be the last thing that we do today. We might end over the next two slides here. I want you on your paper, see if you can put these particular steps that involve the start of a muscle contraction to an end of a muscle contraction. So we've got here 7 steps. See if you can write these down. If you want to abbreviate on your notebook paper, you can right now. Try to put these 7 steps in order from the initiation of a muscle contraction and then ultimately to the stopping of a muscle contraction where we have something that's going to have the contraction in. So we know contraction ends is going to be, that's our last step. But for these other 7, I want to give you about 2 or 3 minutes. See if you can do this. We'll go over the answer. It'll be the last thing that we do today. We'll do this over the last 4 or 5 minutes of class. This is not for points. This is just for you learning. Rita.
What about binding sites on actin or blood? Do you want that person to be
I don't. That's going to be in the end. That's not going to be your first one. So assume don't start from the first event that's going to initiate a muscle contraction. What's the first thing that's going to start a muscle contraction? It's going to occur during a muscle contraction. So don't start with the resting state, so to speak. That's a good question. I hadn't thought about it that way. And this also points out ideas for study. Do you do things like this to study? Do you actually use writing as part of your study? Now, all the possible steps are not there. We could add more steps in there, but I have put just a certain number of steps. Could you order those? You ever get with a study group and maybe design puzzles like this for each other? If you want to work with somebody next to you right now, that's fine. If you want to talk, that's okay. That's a part of your learning strategy. If you've got part of it and kind of stuck on a particular area, you want to consult with somebody nearby, that's fine. Okay, we'll go over the answer here in just about 1 minute or 2 minutes. It'll be the last thing we do. Lorena?
Didn't the third one down be the first step or the last, or the fourth step?
Binding sites on actin or block, that one you're asking at?
Yeah, could it be the first or the third?
That's what Rion asked over here. That's not going to be your first step in this one.
Okay.
So I want your first thing to be, what is the initiation that causes contraction?
Gotcha.
So I mean, that could be the resting state, would it exist before contraction starts? But I guess that's a good question, and again, Rion just asked that. So we're going to start, what's the first thing that's going to happen in order for contraction to occur?
The ACH. The second one being muscle
Again,
action potential.
You may not have all possible steps that we could put in there, but among these, can you put these in order? Okay, we've only got about 1 minute left in class, so let's go ahead and see how far you got. You may not have got all of these, but let's see how far you got. By the way, if you haven't done anything, shame on you, at least try. Let's go ahead and do the steps here. What's the first thing you came up with?
ACH release.
How about ACH release from synaptic end bulb of axon? So that's the first thing. That's acetylcholine, release from synaptic bulb of that. What's the next thing on our list here?
Axon potential spreads across sarcolemma.
Okay, number 2 would be muscular action potential spreads across the sarcolemma. I mean, I've not got all possible steps in here, but among these, that would be our next one. What happens next? Okay, number 3 would be calcium released. So calcium is released from sarcoplasmic reticulum. What would be the next step? Okay, calcium binds troponin. That makes tropomyosin shift off away from the active sites. So now our active sites are exposed on actin. So now what happens?
Myosin hits.
Myosin heads bind to actin and then pulls, assuming there's ATP present, which there should always be. And I think we're going to have one last step now. Is that right?
Binding sites are blocked.
Okay, so what happens when, I guess the next one we're going to have eventually is binding sites on actin are blocked from, presumably we've got a stoppage of the whole system. If you didn't get all of that, it's on the next slide. So if you didn't get all that written down, it is on the next slide.